Hello students, this is Dr. M. Bhushanam, Associate Professor in the Department of Zoology, Maharani Science College for Women, Bengaluru. Students, take care of your health during this pandemic time. In today's session, we shall learn another topic of genetics, that is, inheritance of continuous characteristics or polygenic inheritance. We also call it as inheritance of continuous traits. So this concept is important for essay question. Objective of today's session is to learn the polygenic inheritance with reference to the skin color in man. The content includes polygenic inheritance in the skin color of man. Well, students, we know that according to Mendel, every trait and character is controlled by a single gene. So each gene is represented with two number of alleles and such traits are generally referred as Mendelian traits. Down in the picture, when we look at the classical example of Mendel related to the pea plant, when a tall pure line of pea plant is crossed with a dwarf pure line plant, that is a homozygous dominant crossed with homozygous recessive, you find only two alleles representing a single trait, and hence in F1 generation you find all tall character plants. So this kind of pattern of inheritance is called Mendelian inheritance, and such traits are called Mendelian traits. But remember, all the traits do not follow the Mendelian pattern of inheritance with two alleles or one gene. According to a scientist by name Mather, there are many genes that can influence or control collectively the character. I mean to say single character. So when many genes control a single trait, they are called as polygenes, and the pattern of inheritance we call it as polygenic inheritance. I want you all to recollect back your memory. We know that Mendel has called genes as factors. So when polygenes control a character, poly means many, genes here referring to the factors. So multiple factors will involve in this kind of inheritance pattern. So in polygenic inheritance, we generally find several genes which will collectively affect the quantity of a character. So many genes will influence ultimately the character of an organism. So here in the picture, when we look at the same classical example is the skin color in man. We find here three dominant genes of A, B and C controls the character of skin color in man. So to brief the content, the number of dominant alleles of these three genes will control a phenotype in the skin color of man. I mean to say, as the number of dominant alleles vary, the phenotype of the individual also starts varying. So this is what we call it as the multiple factor inheritance or polygenic inheritance. So this kind of inheritance pattern, since it is related to the number of genes and the additive effect, the additional effect given by each of the dominant allele, we call the condition as, or inheritance pattern as, quantitative inheritance. So quantity here refers to the number of alleles. So number of dominant alleles will control the character of the phenotype in the individual. And when we look at the genes, every gene will be independent of their expression, and at the same time they add up to the character of the same. So all genes will express the character equally, and each of which, when are added to it, say suppose if we have A, B and C, all the 6 are dominant alleles. A dominant alleles will have one effect. Similar effect will be shown by B alleles. Similarly, that of the C alleles. When A is getting added to B and C, the effect becomes more. It is like we adding or boosting up the effect on the organism. So the phenotype of the organisms totally changes as the number of dominant allele gets added up. And this is hence is called as continuous variation. So continuous traits, otherwise referring to the polygenic traits. Reason is that we find addition of every dominant allele will add up a new character into the organism, even though they influence a single trait. So that's the importance of quantitative inheritance or continuous variation. The examples pertaining to this is height and weight in human beings and skin color in human beings. It was Yule, Nilsson-Ehle and East were the group of scientists who worked on the pattern of quantitative variation and inheritance that are controlled by a large number of individual genes. So, the polygenic system that involves many number of alleles and genes controlling a particular character is explained by the scientist group of Yule, Nilsson and East. Students, remember, this concept of polygenic system is related directly to the multiple factor hypothesis. So what is this multiple factor hypothesis is of? The hypothesis states that for a given quantitative trait, a trait that follows quantitative inheritance, in simple, a trait that is influenced by many number of genes, several genes, which are independent in their segregation. So when they move during the segregation process, they are independent and they will show an additive or cumulative effect on the phenotype. So just to give you a brief idea about it, when you look at the color variation in the checker box that is given down towards the left side, you find number 6 representation of the block seems to be very dark. So it indicates that 6 dominant alleles are there, indicating that particular coloration. And 5 indicates a particular brown color. Can you see that? So that indicates that the effect is little lesser compared to the 6 numbers. And 4, it is much more lesser. 3, much more lesser. 2, much more lesser. 1, much lesser. And 0, no effect at all. So you can find the color between white, where 0 is seen, till 6, where the dark black color is, block is seen. We find the variation in the color. So number of dominant alleles will affect the phenotype of the individual. So cumulative effect of these genes are seen on the phenotype. So this is what we call it as multifactor inheritance or polygenic inheritance. So to understand this polygenic inheritance, a very classical example that we know is skin color in man, which was given by scientist Davenport. So down in the picture, you find towards left side the coloration of the skin color change, and towards the right side is the photo of Davenport, who gave the concept of skin color in man. Well, students, now we shall understand the characteristic features of polygenic inheritance. When can you call the inheritance pattern as polygenic inheritance or multifactor inheritance? The first point here, we need to define the concept of polygenes. What are polygenes? Poly means many, genes are factors. So many factors or many genes that influence a single character or single phenotype is referring to polygenes. So this is what point number 1 explains. A gene that employs minor effect on the phenotype along with the other genes is referred to as polygenes. So in simple, when many genes influence a single phenotypic trait, we call it as polygenic trait. The second point is the effect of every individual gene is very minor or very less, but it is added to the cumulative effect, overall effect of the phenotype. So the effect of every gene is equal here. Equal effect is seen, and every gene's equal effect is added to the expression of phenotype of the individual. Down in the picture, when you look at 0, when you see, 6 number down when you see towards the extreme right side, that is the total number of dominant alleles, indicating a very dark complexion or a black colored complexion or negroid complexion. As the number of dominant alleles reduces from right to left, the color change is seen in the skin color of man, indicating that every gene that is added there is adding up the equal effect and additional effect it is of. The next important feature is that every individual allele will contribute to the cumulative or additive effect on the phenotype. So expression of every gene, when you look at every allele of a gene, when you look at it, is not controlled or it is not masked, it is not covered. So the concept of dominancy doesn't play a role here. I mean to say, when 2 dominant genes are there, both will show its expression. When 4 dominant alleles are there, all the 4 will show its expression. So the effect is equal and it is cumulative, and the effect of one allele is not masked by the other one. So this kind of additional effect or additive effect shown to the phenotype of the individual will show continuous variation. Students, remember, the term variation refers to change in the trait. So it is the change in the character keeps changing continuously for the phenotype when the dominant alleles increases. So that's what the color change that you find in the picture that's given down. When one allele is there as dominant, the color is different. Two are there, the color is much different or much darker. When 3 are there, much more darker. When 4 are there, much darker. 5, much more darker. And 6, that is the darkest condition. So this is what we call it as continuous variation, continuous change in the phenotype. It is added by every dominant allele. And it is the pattern of inheritance when we look at of polygenic trait, it is very, very difficult to predict. So when you look at number 4 dominant alleles of the checkerboard there, they all show similar pattern. When we look at 3, they all show similar pattern of the skin color. 2 similar, 1 similar, so 0 similar, 6 similar, 5 similar. So you find for every number a similar color is quoted, indicating that they all resemble same. But the inheritance pattern, when we look at it, cannot be determined that easily. The next important feature is that polygenic inheritance, when is drawn on a normal distribution, the curve appears to be a parabolic curve, where the intermediate will occupy the maximum. That is, number 3 individuals are maximum. Next, number 4 and 2 are equal on either sides, and 1 and 5 are equal, 0 and 6 are equal. So when you look at the bar graph that is shown beside this checkerboard, the middle color indicates that it is the intermediate individuals. So all the time, intermediate individuals will have maximum number in the phenotypic condition, and least is followed by the 2 extremities. Students, that one aspect. The second important aspect is that besides the middle range, when you look at, they are equal. For example, middle one is 3 here, and besides of it on either side is, so one is 2, the other side is 4. They are equal. 1 and 5, they are equal. 0 and 6, they are equal. So equality is seen or stability is seen on either sides of the middle value. So this is what we call it as normal distribution curve. When you look at the number, say suppose if 3 individuals who have occupied at the center are 20, the next 2 sides of either sides of the middle region, when you look at, it is for 4 and 2. So they will be of 15 individuals. Then when you look at 1 and 5, they are around 6 on either side, and 1 individual of 6 dominant alleles and 1 individual with no dominant alleles. They are also equal on either side. So 1 is to 6 is to 15 is to 20. That is to 15, then is to 6, then is to 1. So stability is seen on either side. This is what is the importance of normal distribution curve. The next important point is that polygenic inheritance is different from that of multiple alleles. Students, remember, the concept of multiple alleles indicates that many alleles of the same gene will control a character. So they occupy same genic locus on the chromosomes. So these alleles are many in number, they control a particular character. But if many genes control a single character, we call it as polygenic inheritance. So polygenic inheritance is totally different from that of multiple allelism. So polygenic inheritance will lead to a newer branch in the genetics called quantitative genetics or biometrical genetics, where it refers to measurement of the values. As the number increases, the character keeps changing. So that's the specialty related to polygenic inheritance. So with this we are completing the importance of characteristic features related to polygenic inheritance. Students, after we understanding what is polygenic inheritance is and the features related to it, the continuous traits. Now we shall concentrate on the example of inheritance of skin color in man. As we have discussed earlier, it's a classical example explained by Davenport in 1913 from Jamaica, US. So down in the picture towards the right side is the photo of Davenport. So he is the one who explained the inheritance of skin color in man, what we are going to understand now. According to the scientists, there are 3 genes, that is 6 alleles that controls the skin color in man. So these 6 alleles, if are in the dominant condition, such as for A gene capital A, for B gene capital B, for C gene capital C, capital C. If all the 3 genes with 6 dominant alleles, if are, they control the character of dark complexion of the skin. So each genic allele will add up equal and similar effect onto the phenotype. So that's maximum coloration of the skin is seen. So melanin synthesis by tyrosinase enzyme is to the maximum condition. So this condition we call it as dark skin or black skin. When all the 6 alleles of the 3 genes are in recessive form, they form a color called white. So no melanin synthesis, albino otherwise. So negroic individuals are black, white individuals are albino. When these 2 conditions are seen, when all the 6 dominant alleles are seen, it forms a black color, and if all the 6 alleles are in the recessive condition, they form albino color. Now, when this genotypic individuals, one with pure dominant genes and one with pure recessive genes, that is an American white and a Negroic African individual, when they are crossed, we tend to find an F1 offspring who shows the character of 3 dominant alleles in them, 3 recessive alleles in them. So alleles of dominant and recessive are equal. When 3 dominant alleles are seen, the color of the skin is said to be intermediate, and it is also termed as mulatto. So mulatto color is the intermediate color skin, which is seen when 3 dominant alleles are there in the individual. So 3 important aspects we have learned here, students. For all the 6 dominant alleles are there, we find negroic individual color. So if all the 6 alleles are recessive, we find white or albino color. When you find 3 dominant alleles, we find the color called intermediate skin color or mulatto. Now, so this is what in F1 generation that you find in the picture down. So when you look at here, the cross, the coloration of the skin, when 6 dominant alleles are there towards extreme right, you have color of the black. When all the alleles are recessive, you find white, and intermediate color with the 3 dominant alleles is mulatto or intermediate skin color. Now, to have F2 generation, we are going to cross 2 mulattoes of F1 generation. So when F1 generation individuals are crossed, we tend to find F2 generation where the color of the skin will be intermediate between albino or white to mulatto, then from mulatto to dark. So that's the color of the skin that we find in F2 generation. So as we have understood, let us now understand it by cross. We know when white individuals are crossed with black, in F1 generation we find mulattoes or intermediate. Students, remember, these intermediate individuals, when are self-crossed, the genotype representation of mulatto is capital A small a, capital B small b, capital C small c. When we find this kind of genotype, how are we going to represent the gametes? To understand this, first we are going to separate out capital A and small a as it is shown in the left side of the figure in between. So we have capital A, small a separable. Then to each cap of the A, we are going to separate B. So A will join with capital B, small b. Small a will join with capital B, small b as represented in the picture there. Now, each of B will have again combination with C. So that is one capital C, small c. When you look at here, capital A will have option of joining with capital B, small b. Capital B will have option of joining with capital C, small c. Similarly, small b will have option of joining with capital C, small c. So likewise, when you look at for small a, we have capital B, small b. Each of the B will have combinations with the capital C, small c. So altogether, when we look at to represent the genotype of the gametes, we have capital A, capital B, capital C, the first gamete. The second gamete is capital A, capital B, small c. The third gamete is capital A, small b, capital C. The fourth one is capital A, small b, small c. So 4 gametes are formed from the first set of capital A. Now with small a, when you look at small a, capital B, capital C, one set. Small a, capital B, small c, the second. Small a, small b, capital C, the next one. Small a, small b, small c is the last one. So down when you look at, we have 8 different types of gametes that are formed from this genotype at one side. So when an intermediate individual is forming the gametes, how many types of gametes are possible? It is 8 types. So similarly, from the other individual, we have another 8 gametes. So 8 into 8, 64 gametes are formed. So here is the picture that represents again the gamete formation as I have explained you. Capital A, capital B, capital C, one set. Capital A, capital B, small c, second. Capital A, small b, capital C, the next. Capital A, small b, small c is the fourth one. So that forms 4 sets of the gametes. Next, small a, capital B, capital C, the second. Next, small a, capital B, small c, the next. Small a, small b, capital C, the next one. Small a, small b, small c is the last one. So 8 different types of gametes are possible. So this is how it is easier for you to represent the gamete production for the genotype, whatever it is of. So when 3 genes are involved of intermediate type, this is how the gamete representation will be of. Now, so when you represent 8 into 8 gametes in the checkerboard, we find 64 offsprings. So you just see the cross, and after you making the permutation of the cross, you try to write the total number of dominant alleles that are there in each of this box or block. So that will show after writing. One of the 64 will be Negroic with 6 dominant alleles. 6 of 64 individuals will have very dark skin color complexion with 5 dominant alleles. Then 15 of the 64 will show dark color skin complexion showing 4 dominant alleles, and 20 of 64 will show mulatto or intermediate skin color with 3 dominant alleles. Now the same will repeat for the further. 15 of 64 will be fairer, with showing 2 dominant alleles. 6 of 64 will show very fair skin showing 1 dominant allele, and 1 of 64 will show white or albino color where there is no dominant allele. So when you look at the cross, 1:6:15 is to 20. Now repetition on either side, 15:6:1. So it is like when you draw a graph, it will show a parabola. So that's the importance. So what's the resultant phenotypic cross of polygenic trait? It is 1:6:15:20:15:6:1, and this ratio will hold good when there are 3 genes control a character of polygenic inheritance. So if it is 2, the ratio will vary. So for 3 polygenes, so this is the ratio that you find. Remember how to know that it's a polygenic trait. It is easily seen when there is equal distribution on the individuals on either side. So stability is seen for the ratio. So here is a picture that represents the color directly as what we have understood earlier. So with that, we are completing the concept related to polygenic inheritance, students. Let's now understand and answer MCQ questions related to the polygenic inheritance. Quantitative variation is controlled by large number of individual genes are called as what type? It is polygenic system B. A classical example of polygenic inheritance in skin color of man is given by Davenport. Answer is A. So the classical example of polygenic inheritance is skin color in human. So answer is C. The number of genes of true Negro skin color, that is dark black color, in human are, what are the genes that represents dark color of the skin? It is capital A, capital A, capital B, capital B, capital C, capital C. So answer is D. When a mating of 2 mulattoes, that is intermediate skin color, is seen, that produces variety of skin color in the offsprings. What is the phenotypic ratio that it forms? As we have understood, there should be equal distribution on either side with a maximum value at the center. So answer should be perfectly, since 3 dominant genes are involved in the cross, the ratio is capital See answer. 1 is to 6 is to 15 is to 20. 15 is to 6 is to 1. So answer is D. Well, students, what is the outcome of today's session? We have learnt the polygenic inheritance in the skin color of man. How many genes will influence the skin color in the human beings. So references includes the web references of Wikipedia and Britannica.com. And book references includes a textbook of genetics by Ajay Paul, genetics by P.K. Gupta, Introductory Zoology Volume 5 by C.M. Vimala, and Principles of Genetics by B.D. Singh. Thank you all, students.